Okay, hello again. My name is Juthapa Pongklaokam. Thank you for having me here. I would like to present my proceeding in investigation of FOXO3A, serine 253 phosphorylation FOXO3A and 1433 expression in androgenetic alopecia versus normal, supervised by Assistant Professor Lanyu Panikorn. For an introduction, the pathophysiology of androgenetic alopecia are mainly focused on genetics, androgen, microinflammation, oxidative stress. Ultimately, this factor result in stem cell apoptosis, follicular fibrosis, collagen induction, and hair miniaturization. Currently, the FDA approval topical and oral drugs are effective, but only just in the mild and moderate cases, and also have various side effects, could be regularly observed among the treatments. More importantly, a discovery of new competent pathophysiology of AGA could pave a new promising way to understand new effective treatments. Typically, hair follicle stem cells residing in the follicle bulge interact with mesenchymal stem cells and the dermal cells, causing cell proliferation toward hair regeneration by inhibiting inhibitory effects. Hair follicle stem cells are maintained in quiescent state until they receive the signal and then migrate either to the dermal papilla and to the epidermis in the basal layer. Androgenetic alopecia could be associated with the hair follicle stem cell progenitor cell deprivation. The papilla cell from the balding scalp of AGA patient also exhibit signs of senescent characteristic. And from the study in 2015, they also show the premature senescence in response to the oxidative stress compared to occipital dermal papilla cells. The relationships between autophagy and hair biology was found in the requirement of autophagy for androgen induction and proper hair growth, the role of autophagy in cell senescence induced by persistent WIN1 expression of hair follicles, and autophagy promoters and inhibition of autophagy inhibitor promote hair growth. For example, in 2021, the researchers took 6 hair follicle units for hair follicle culture from donor site area of 3 AGA patients. The results show that an inhibition of autophagy, especially from the initial step and by an interaction between BCL1 and BCL2, leading to hair miniaturization by enhancing premature collagen and cell apoptosis. Moreover, in 2021, the study demonstrated that myasteric acid is an effective promoter of anagen signaling by autophagy through an activation of Wnt beta-catenin and ERK pathway in the dermal papilla cell, leading to the dermal papilla cell proliferation. FOXO can regulate autophagy via transcriptional, post-translational, and epigenetic pathway. As translational factor, FOXO activate autophagy genes by binding to the promoters to induce autophagy in multiple stages. At the post-translational level, FOXO may interact with autophagy-related gene 7 protein in the cytoplasm to induce autophagy as well. And from the previous knowledge, active AKT translocate into the nucleus and phosphorylate FOXO3A at 3 conserved region residue, including serine 2 and 53, generating a binding site for 1433 protein, causing FOXO3A nucleocytoplasmic translocation and eventually cytoplasmic degradation. In conclusion, these findings lead to our research question whether there is a clinical pathological significant difference in the expression of FOXO3A serine 253 phosphorylation FOXO3A and 1433 protein between the AGA group and the normal group. Could FOXO3 be the key regulator of autophagy in AGA, which could lead to the new promising etiology of AGA treatment? Our study is aimed to determine the clinical pathological significance of immunohistochemical expression of the FOXO3A phosphorylation, FOXO3A, and 1433 expression in the outer root teeth of bowel region, comparing between early stage of AGA and the normal donor. We divided our population into 2 groups, including 2 normal healthy volunteers and 3 early onset AGA patients. The inclusion criteria of androgenetic alopecia are male patients aged between 20 to 40 years old with hair miniaturization more than 20% at the vertex and less than 20% at the occipital scalp, with the presentation of bowel polypilasi, which is the early sign of the AGA, and the physical examination showing Hamilton Norwood state 2 to 3 at the vertex type. And other treatments of hair loss, other relevant comorbidities and disease are excluded. 5 micromillimeters or 5 micron formalin fixed paraffin embed vertical section of biopsy scalp samples were prepared for H&E staining to identify hair follicle. Then staining slides were carefully selected from H&E staining slides. We identify each area of hair follicle from morphology, concentrating in the bulge area of the outer root teeth where the stem cell residing. We recruited multiple bulge areas acquired from 6 tissue sections of AGA group and 4 tissue sections from the normal group for each staining. And automated immunohistochemical assays were carried out in this study. Immunohistochemical assays were performed on Ventana ultraview depth detection kit in Ventana benchmark XT stainer. And these are the antibody using in our immunohistochem staining and its positive control. Digital images of immunohistochem stain slide were obtained by slide scanner and then uploaded into the server. The scanned images were uploaded into QPath program and manually selected the interesting area within 300 wide and 300 range micron, including the bulge area of the outer root teeth and excluded area represented with artifacts. Immunohistochemical staining was assessed and scored by using QPath software, the health score method of scoring, and also the percentage of total positive was applied for the evaluations. The SPSS was used to statistical analysis, and we used the non-parametric test, Mann-Whitney U test for analyze the information. Okay, for the result, the normal group show normal hair and scalp with the dermoscopic features, showing hair follicle units containing 2 to 4 terminal hair with less than 20% of miniaturization. Androgenetic alopecia have the dermoscopic examination presented with miniaturization more than 20% at the vertex scalp and also the brow polypilasi, like in the back arrow I show you in this slide. And here are the original images of the outer root sheath bowl stained with FOXO3A on the left side with A in AGA group and C in the normal group. And on the right side of the picture are the markup images showing cell detection and classification using QPath. The red represented the 3, like the 3+ intensity, the dark or the orange color represent the 2+ intensity, and the the bright yellow one represent the 1+ intensity, and whereas the blue one represent the negative one. Although his goal and the percentage of total positive of FOXO3A among ACA group are much higher than the normal group at the median value of 254.14, and also for the total positive with 98.97%, these variables were not statistically significant difference. And moving to the next staining, the phosphorylated form of FOXO3A, we found that we found a significant difference between two groups, with the higher level of the median score in the AGA group at the median value of 27.16, and for the total positive at 19.83%. As for the 14-3 protein compared between two groups, there were not significant difference. The discussion. Our finding show significant evaluation of phosphorylated form of FOXO3A among the AGA group at the bulge area of the outer root teeth. Sorry, the slide will not go. Okay. And this is the first study of FOXO3A and its related proteins in hair disease. Here we saw the significant difference of phosphorylated form of FOXO3A in the bulge area of outer root teeth among the AGA group, which is presumably represent the binding of this molecule with 14-3 protein, resulting in FOXO3A being excluded from the nucleus, sustained in the cytosol, and gradually degraded. Therefore, reduction of an active form of FOXO3A in the nucleus led to poor transcriptional activities regarding the autophagy-related genes. However, an overall FOXO3A protein expression was not statistically significant higher in the AGA group than in the normal group, which might caused by an accumulation of cytoplasmic FOXO3A and in an active form. We hypothesized that increased expression of serine 253 phosphorylation FOXO3A in the AGA group could indicate that an imbalance of this molecule involved in the impairment of cellular homeostasis, particularly autophagy in stem cells located in the bulge area of outer root teeth, which is responsible for anagen induction, collagen inhibition, and dermal cell proliferation, leading to clinical manifestations of hair miniaturization among AGA. Limitation is that FOXO3A has potential to protect stem cells through mechanisms other than just autophagy-regulated genes. Target genes of MPK FOXO3A pathway, for example, are also involved in defense against the oxidative stress and also the DNA damage, and also the small sample size. In conclusion, this is, to our concern, the first study demonstrating the clinical pathological significance of early onset AGA and the imbalance in cell signaling of the protein involved in autophagy, including FOXO3A, serine 253 phosphorylation FOXO3A, and 14-3. Phosphorylation form of FOXO3A was found to be significantly increased among the AGA group in the bulge area of outer root teeth region, which could be the potential key molecule to correct dysregulation of autophagy in AGA. However, further investigation should be conducted. We propose additional immunofluorescence study to localize the serine 253 phosphorylation form of FOXO3A in the nucleus, as well as their localization with FOXO3A and other autophagy-related proteins to determine the interaction of the FOXO3A and autophagic flux protein in both groups. Thank you for your attention. And any question from the floor?
Okay, thank you. I would like to ask how you're gonna to apply your findings to the patient for the treatment or whatever.
Okay, so according to our finding, we assume that FOXO3A had an effect on AGA as the transcriptional factor to produce the protein that involved in the inhibitory effect of deminiaturization. So I think if we can determine which we can determine the target of this process, we can produce new treatment or new drug that emphasize on the autophagy or emphasize on the increase of FOXO3A level in order to protect cells from any stress, any cellular stress such as oxidative stress or DNA damage or cell senescence, which usually occur among the ACA patients.
Thank you. Any more questions from the floor? Audience.
Can you give some more examples of the diseases besides the alopecia that you mentioned? Some other systemic diseases that has some defect about FOXO3.
Okay. Mainly FOXO1 and FOXO3 has a very study in skin. Particularly FOXO3A have the function to protect cell against the melanoma from cell cycle regulation. And as for FOXO1, it has a role in acne formation and also the skin morphology.
Okay, thank you. Thank you very much.
Any more questions? Okay, so thank you, Dr. Juthapa.
Thank you,ค่ะ